Hi everyone, my name is Lisa Janssens and I'm a second year PhD student at the Lab of Toxicology in Ghent. I work on assays to sense HIF stabilization. And when you hear HIF stabilization in a doping context, you would probably think of the newly emerging HIF stabilizers that doping control is encountering. And that's indeed what I want to talk about. With taking HIF stabilizers, you could say that athletes try to mimic a state of hypoxia even more pronounced than when training at high altitude. The result is comparable. The body produces more EPO and therefore more red blood cells. To show you why that is and how we can use this to our advantage in doping control, I'd like to zoom in part by part of my poster. Let's start with the stabilization part. HIF is a transcription factor, which means that it modulates the expression of genes. But HIF consists of two subunits, and the alpha subunit is normally degraded constantly. And this degradation process concerns both principal isoforms, HIF1 alpha and HIF2 alpha. Now, stabilization of HIF means that you inhibit this degradation process by inhibiting the responsible enzyme, which creates a comparable effect as when less oxygen is present. HIF alpha will accumulate and will interact with the beta subunit to form and act as a functional transcription factor. This then results in gene expression and in the production of, for example, EPO. Multiple HIF stabilizers have been developed with highly varying structures. And this complicates the structure-based detection, especially as new compounds are constantly emerging or being investigated. But the thing is, they do have one thing in common. They all inhibit the enzyme responsible for HIF degradation and therefore they stabilize HIF. Now, why don't we use this action as a method for detection? Well, we did. And we did so by fusing the different subunits of the HIF alpha, of the HIF1 and HIF2 transcription factor to different parts of a split luciferase. And we introduced these fusion proteins into cells. When we then treat the cells with HIF stabilizer, HIF alpha will accumulate, and the more HIF alpha is present, the more it will interact with HIF beta and form the functional transcription factor. But if you look closely, you will see that this interaction also brings the split parts of the luciferase closely together, and therefore the enzyme will become functional again. When we then add a substrate, the enzyme will produce light indicating that interaction happened. And this light can then be monitored over a period of time. When we then look at the outcome of this assay, you'll see that it clearly distinguishes signals from cells incubated with HIF stabilizer over cells incubated with only solvent control. We tested the performance of the assay in more depth, and we saw that we can generate concentration response curves of the different HIF stabilizers. But more importantly, we already saw, or we could already improve the sensitivity compared to the state of the art in this field. You can see this by looking at the different scaling of the graphs and by looking at the different concentrations that could be detected by newly developed assays but could not be detected by the state of the art. Now, wouldn't it be great if we can optimize the sensitivity even further in order to apply these assays on biofluids from athletes? We hope that this assay can serve as a screening tool to detect all HIF stabilizers at once, current and future doping substances. Because let me ask you, why detect one compound based on its structure when you can detect them all based on activity? I would like to thank the PCC for their trust and financial support for this research, and I hope you agree that sensing HIF stabilization itself as a method for detection could be the start of a new approach in screening for HIF stabilizers. Because then we would have a method that's here to stay and that will pick up every substance that induces HIF, current and future ones.